Today we'll talk pretty much about one study, and this is a recent report that came out in the New England Journal of Medicine on the efficacy of remdesivir. And I think it's an essential skill for you all to be able to read the primary literature on this topic and make a decision for yourself what evidence is actually there to support a particular course of treatment. And then we'll start talking a little bit about drug resistance. Drug resistance in COVID and SARS-CoV-2 isn't a thing yet, but it probably will be as we treat it more. So I'll switch a little bit. In addition to talking about SARS-CoV-2, we'll also talk a little bit about bacterial pathogens. So what is the state of the art, so to speak, in COVID-19 treatment in the United States? If you go to an NIH webpage, which is listed here at the bottom, the link is listed here at the bottom. This is the current recommendation of what should be done depending on the severity of the case. So, for example, if disease severity sort of goes into these four broad categories, so mild, and then progressively more severe. So in the blue bin, you see not hospitalized or hospitalized, but does not require supplemental oxygen. The current recommendation of the panel is to basically not treat with anything specific antiviral or immunomodulatory. So, because a lot of, prominent government figures were recently infected with COVID, particularly President Trump, he was treated with pretty much everything that's available in the arsenal at the moment. We'll talk about remdesivir, but when people talk about immunomodulatory therapy, something like Regeneron monoclonals will fall into this category. We'll talk about them next time. And pretty much the panel recommends to not do anything in special at this point. And then if you see, when they make a recommendation against, say, the use of dexamethasone, which is a corticosteroid, they cite evidence called A1. And if you look at the bottom, the rating of recommendation, A means is strong, B moderate, and C optional. And then they base this on a one or more randomized trial of clinical outcomes and or validated laboratory endpoints. Then moving down the line, if you're hospitalized and require supplemental oxygen, but does not require oxygen delivery through a high flow device, so basically no ventilator, then the recommendation is to either do remdesivir or remdesivir plus dexamethasone, and this kind of delivery. The same thing for hospitalized and requires oxygen delivery through a high flow device. And then finally, for the most extreme case, there is a different recommendation. Pretty much you only see two drugs appear on that list. So remdesivir, which is we'll talk about today, and dexamethasone, which is just a corticosteroid. It has nothing special in its action that is targeted towards COVID-19 or SARS-CoV-2. So if you dig a little bit deeper into this website, you will not actually find that much. In fact, there's a disclaimer at the top of the site that states, quote, there are no Food and Drug Administration approved drugs for the treatment of COVID-19. So pretty much everything we are using today is either compassionate use or, off-label or just general, not COVID specific. There are sort of three categories of this of the therapy that you can consider. So you have antiviral drugs. So this is something that is designed to directly or indirectly act against the virus. Right. So this is a specific medication or compound that's designed to interfere, say, with viral replication or some other life cycle and directly attack it. The second category of therapy would be immune therapy. And this is fairly broad, so something like convalescent sera from patients that recovered or monoclonal antibodies would go into this. And this also includes just general immune modulatory compounds. And then you have this broad category of what's called adjunctive therapy. So anything that is not directly attacking the drug or stimulating the immune system. So things like vitamins, vitamin D, or antithrombotic agents, so against blood clotting, and dexamethasone, which is a really old drug. So first approved in 1961, is just a generic anti-inflammatory corticosteroid. So not a whole lot in the arsenal at the moment. And as far if you dig into the antiviral drugs that have been sort of listed on the same webpage, there are, so one of the things, they're all repurposed drugs, right? So none of them were designed specifically for the virus at hand. So remdesivir is the closest, and the plus sign means that the panel recommends its use, as you've seen before. The three other drugs that are being used that have sort of received a significant amount of coverage and controversy would be hydroxychloroquine, plus azithromycin, which is just a generic antibiotic. Hydroxychloroquine is an antimalarial drug. So the panel, based on the current available evidence, does not recommend its use. Lopinavir or ritonavir and other HIV protease inhibitors, as it says in the name, these are drugs designed to treat HIV, so that they were used early, especially in China. There were early reports that they did something. Again, there's at the moment no evidence that that's the case. Or ivermectin, which is an antiparasitic medication, also not recommended. So what all this recommended or not recommended goes, this is all based, at least in the United States, on fairly strict definitions of what is the minimum required evidence in order for a particular treatment to be approved or recommended. And the gold standard for evidence-based medicine, which is what is practiced in the United States, is the randomized clinical trial, right? So the cartoon, of what that is here, and it's very, very simple. But of course, nothing is as simple as this. But a clinical trial, you all have randomized clinical trial, has a very intuitive concept, which is you take your sample population, you split it into two or more groups, and you do it randomly. Although that is also not as simple as it seems, as you'll see in the remdesivir trial. And then you separate the individuals that are in the study into two arms. Group one, which is treated, so you treat them, you observe the outcome. Group two, you don't treat them, or you either treat them with a placebo or you treat them with a control treatment, whatever that might be, the standard of care, and then measure the outcome. And the randomized control trial, which is abbreviated as RCT, is a trial in which subjects are randomly assigned to one or two groups. One is the experimental group, which is receiving the intervention that is being tested, and the other, the comparison or control group, which is receiving an alternative, conventional treatment or placebo. The two groups are then followed to see if there are any differences between them in outcome. The results from subsequent analysis of the trials are used to assess the effectiveness of the intervention, which is the extent to which a treatment, procedure, or service does patients more good than harm. RCTs are the most stringent way of determining whether a cause-effect relationship exists between the intervention and the outcome. So the key word here is cause-effect relation. And this is why, what we're really measuring. So we're going to spend a little bit of time just discussing sort of some of the fundamentals of why randomized control or randomized clinical trials are an essential tool in establishing this cause-effect relation instead of just an association relation. We'll work through a couple of examples, and I've decided you might want to go back to this at your leisure and examine it. There's a little bit of very, very basic math and probability here, just to define the concepts, and I'll begin by sort of giving you a counterexample just to drive home the point that association is not the same as causation, but in this case, in the context of treatment. So the goal of any trial is to estimate what's called an average treatment effect, or ACE, or sorry, ATE. I can't abbreviate correctly. And the average treatment effect would exactly be as you would imagine. It's, what the application of this treatment would do to an average individual in the population. And how the population is defined would be different, based on what treatment it is, right? So it could be the general population, for example. It could be, say, only women if it has to do something with, pregnancy, for instance. It could be kids. It could be old people. So what the population means is different in the treatment. So let's consider a very simple case where we measure, three things. So we basically look at the entire population. We have a treatment, which is binary, yes or no. And we have the outcome, which is also binary, yes or no. So in this case, it would be, was it effective or was it not effective? So it's fairly abstract. So you have the universe, where there are 8 possibilities where, you pick an individual. That individual is, falls into one of the categories. It's men or women. Then, conditioned in this, in addition to this, you choose whether or not they receive treatment. And in addition to this, you record whether or not they were able to recover, right? Whatever that means. And this is a very simple universe with only 8 outcomes. And you can enumerate sort of the relative probability of each of these outcomes. So this is given by the table here, where you can sort of follow the path. You make the choices successively, and then you repeat, you arrive at the probability of a particular arrangement of variables occurring. So there should be 8 of them because there are 8 possibilities, 2 times 2 times 2. Each of them is assigned a non-zero probability, and the sum of the probabilities should be 1. So for example, you can read this table by saying the probability of a successful outcome, which means y is equal to 1, for a man, which means that z is equal to men, when no treatment is applied, x is equal to 0, is 3.75%. For a woman, the only thing, if you only change the gender, then the probability is very different. So it's 26.25. Question is, where do I find the syllabus for the scores? There is no formal syllabus for the scores, right? It's basically on Canvas, just the lectures. If you have specific questions, you should, contact us on Slack. We've talked a little bit about the paper. There is no, I haven't picked an exact date yet. I mean, we still have 2 months until the semester ends. If you really want to know, I can probably, pick a date. But I if you want to start working on it now, I suppose that it's okay. This example is a little bit contrived, and we saw a few examples of it last time, Simpson's paradox. So what we'll do first is we'll basically sort of simplify the problem and just average out the men and the women, right? So sum over the entire population. Here it's broken down by gender. So let's just compute, what would happen if you've sort of aggregated the effects over both men and women. So you sum over Z. This is called marginalization in statistics. So you would have a marginal XY distribution where you have, so to get this 0.2, you would take, men with 0 and 0 and women with 0 and 0 and add them up. So you get 0.2, 0.3, 0.25, and 0.25, right? So this is now basically, if you give, treatment, presence of treatment, in the absence of treatment, the probability of a failed outcome is 20%. And the probability, if there is no treatment, the probability of a successful outcome or recovery is, 0.3. Right, so you can take this and you can sort of translate this table into something that we actually want to interpret, right? And this is just basic. So there's some terminology here. So P means probability, and the way to read this is the probability of y equals 1, which is the successful outcome, and x equals 0 means there was no treatment. So in English, this means the rate of response in the absence of treatment. So this is 60%. which means if you don't do anything, 60% of the individuals will recover from this condition, whatever it might be. If you treat somebody, which is, you have X equals 1, the rate of response in the presence of treatment is 50%, right? So just looking at this, seems like the treatment is harmful because, the value, there's a better response if you do no treatment. If you break it down by gender, though, you will see that the opposite is true. So now the rate of response in the absence of treatment among men is 30%. So 30% of them will recover if they're untreated. The rate of response in the presence of treatment among men is 40%. The rate of response in the absence of treatment among women is 70%. In the presence of treatment among women is 80%. So it's a classical situation where you sort of, aggregated versus disaggregated. And it's a counterintuitive example to show you that when it comes to treatment and understanding cause and effect, it is easy to construct counterexamples where what you think should hold doesn't hold. For example, here you have a treatment where, for each subpopulation, it is beneficial, but overall for the entire population, it is harmful. Right, so again, just to summarize all of this, so the probability of a recovery, if you treat, is worse or better than the probability of recovery if you don't treat. So treatment is beneficial for men, treatment is beneficial for women, and treatment is harmful overall. Which is, just to summarize all of this again, association is not causation. All right, so all right, so in other words, if you simply so you need to do something more sophisticated to express the relationship that is due exclusively to treatment. When you see bizarre situation like this, there's usually some tricky confounding, which is, for example, the condition, as you may see. As a question, how can we have a situation where the treatment is beneficial for all the subpopulations but harmful for the overall population? Though that has to do with non-random assignment of treatment, right? So this intuitively it usually has, so there's some trickery there in terms of either the treatment is assigned to people that are would have recovered anyway, or there's some significant disbalance between men and women that's not being measured correctly. But it goes back to the idea of confounding, and confounding literally confounding is where you're not really doing a proper comparison. We did this example last time when we talked about kidney stones. So in kidney stones, one treatment was being preferentially assigned to more difficult cases. So it looked like it was failing, even though if you did an overall comparison, it was doing better than the other treatment. So the classic example, that might be easy to understand is, and this is sort of, I don't have it, but when HIV was still, there was no successful cocktail treatment for HIV in the late 1980s. There was the first drug that came on the market was it was called AZT, and it was fairly successful, for a time, even though what it revealed is that you cannot treat rapidly evolving viral populations with a single drug because they escape. But one of the early studies actually demonstrated, quote unquote, that the treatment with that drug was detrimental to HIV individuals. What they forgot to realize is that they were basically giving that drug to the people who were in the worst shape among the entire population. So you sort of make things so difficult for the drug that you're not comparing an outcome that would otherwise be beneficial, as you would expect drugs doing something. This is, again, just sort of basic framework for understanding clinical trials. And this is due to two statisticians, Neyman and Rubin. They came up with it in the 1970s, and this is called the counterfactual model. The counterfactual model, if you've ever seen, like alternative history TV shows like Man in the High Castle, this counterfactual is exactly the same thing. It's like, what would have happened, had something gone a different way? And in this case, it turns out to be a very powerful mechanism for understanding how clinical trials would work. So we'll just consider a very simple case, which is actually not too different from what we'll have to do for remdesivir. Just consider a binary treatment X, so give drug or don't give drug, and a measured response outcome Y, right? So just think recovered or didn't recover. And the whole purpose of a clinical trial is to distinguish the statement that X causes Y, so the reason that somebody recovered is because they were given treatment, from X is associated with Y. So to do so, we have to introduce the concept of potential outcomes, C naught and C1. And C naught is the outcome if the subject is not treated, and C1 is the outcome if the same subject is treated. Right, so if you think about this for a second, it is impossible to both treat and not treat the same individual. So among these two possible outcomes, in real life, you will only observe one. You will either treat the subject or you won't treat the subject. So the outcome that actually occurred is what you observe. The outcome that did not occur is the counterfactual, right? So, and the way to think about it is, you gave this person with COVID remdesivir and they recovered. What would have happened if you had not given them remdesivir, right? They could have recovered or they could have died, but you don't know. And you cannot know directly because you cannot run the experiment. You give them remdesivir and this is it. You can't say what would have happened without remdesivir. But this is a hypothetical, the counterfactual. So you just have to keep it in mind as an abstract idea. So in the treatment Y, could be one of two things. It could be C naught the outcome could be one of two things. It's C naught if you don't treat, or C1 if you don't treat. And as a shortcut, you just write it, Y equals C sub X, right? So, put in 0, 1. This is, So here's an example of what this might look like. So in this case, imagine that C1 and C0 are, say, continuous outcomes, right? So it might be how many days did you spend in the hospital, for instance, or something like that that's measured in continuous numbers. So when treatment is absent, we don't observe C1, right? Because we didn't apply treatment to those individuals, and when treatment is present, we don't observe C naught. And that's why C sub X is called a counterfactual. What would have happened had the treatment been given or withheld? And if we were able to somehow record this pair, so C naught and C1, then we would have all the relevant information about a subject or a case. But we can only ever observe a half, either treated or untreated. So if you look at the table, here are 8 cases. The first 4 did not receive treatment. The second 4 did receive treatment. So for all of them, we measured the outcomes. So, the outcome for subject 1 is 4, say 4 days in the hospital, 7 days in the hospital, 2 days in the hospital, 8 days in the hospital. With the treatment, it's 3, 5, 8, and 9. And you record the outcomes. So in this case, this outcome is the C naught, right? It's what happened if you didn't do treatment. And you do not know what C1 would have been, right? That you don't get to observe it because you did not treat those individuals. And similarly, for the second half, you get to observe C1 because you treated them, but you don't get to observe C naught because you treated them. So you don't directly know what would have happened had they been not treated or treated. So if you think about a binary outcome, right? So survived or didn't survive, or recovered or didn't recover, then you only have 4 possibilities. And depending on what these possibilities are, you will have the following outcomes. So you can have the survivor type that would have recovered regardless of whether or not you treated them or not. On the opposite angle, you would have doomed individuals, so the ones that would have failed to recover or died, either treated or untreated. And then you have the 2 interesting categories. You have responders, which means they would have died without the treatment, but survived with the treatment. And you can also have anti-responders, which would have survived without the treatment, but died with the treatment. It's obvious that for any clinical trial, the only information about efficacy will actually come from the responders or anti-responders, because survivors and doomed individuals give no information. To them, there's basically no effect of the treatment. So what we want to estimate the average causal treatment effect, which is called to theta, and this is hard to estimate, and this is what we want. And the other thing we can estimate is association alpha, which is easy to estimate and can't be confounded. Right, so the standard definition of the average causal effect is if you were able to compute the average response with treatment over the entire population, that's called EC1, and subtract the corresponding average in the absence of treatment, you would get the average effect, right? So for example, if you think about here would be, like rate of survival and so on. So this is impossible to measure directly, because this takes the average over the entire population, and you cannot both treat and not treat the entire population, right? So this is what we want, and this is not something we can estimate directly from the entire population. What we can estimate is this association. We can estimate the association that the treatment is successful if it's applied, minus the probability that the patient recovers without the treatment. Right, so this is something you can directly measure. You just go and count. I gave treatment to this many people, and I gave treatment to this many people, or did not give treatment to this many people. So many people recovered here. So many people recovered here. I have the result. All right, so here, this is the mean outcome if everyone were treated versus if no one were treated. And the information in the clinical trial comes from a subset of individuals that are either responders or anti-responders. So to give you an example of something silly, that would be when association is not equal to causation, or theta does not equal alpha. Let's consider this quote unquote clinical trial on 4 individuals in each. So 8 individuals. So these 4 were not treated. These 4 were treated. For those that were untreated, the outcome was negative. For those that were treated, the outcome is positive. But it just happens so that everybody in this category is doomed, right? So there's no effect of either treatment or not treatment. And say in this category, everybody is a survivor. So in other words, you sort of chose who you treated poorly. So the causal effect here is 0, because you treat everybody on average. So average over the C0 column is 0.5. Average over the C1 column is 0.5. The difference between them is 0. The association is perfect, because the probability of a successful outcome when you have treatment is 1. Everybody who receives treatment is recovered, and everybody who doesn't receive treatment has died. So that looks like it's a great treatment. And it seems like a silly example, but you might imagine it's actually not too silly, because if people basically self-select treatment, this will often happen. So for instance, imagine that you have, Y0 is sick in some sense, and Y1 is healthy. And just assume that healthy people that are already healthy, want to maintain their health, so they take vitamins. And say people that are not healthy, smoke and drink and all the things that you're not supposed to do not take vitamins. So in that case, if only healthy people take vitamins, and only unhealthy people have vitamins, don't take any vitamins, and assuming that in reality vitamins have no effect on the outcome, if it just happens so that healthy people take vitamins and only healthy people take vitamins, there's a very strong association. But it's strongly, and the reason that fails to work is because the treatment is confounded by the health state of the individual, right? So if you just, if you treat all survivors, you'll have a great outcome, right? But you didn't have to treat them, they would have been fine anyway. So it's tempting to interpret association as causation in this case. And for example, if all you do here, if there's still no association whatsoever, but if you just change who takes the vitamins, and now it's everyone except for this one individual. So taking vitamins now makes things worse as more people take it. So, alpha now decreased from, association of 1 to only 4 and 7, right? So that's, if you saw something like this, you would realize that there was something wrong with the original trial, because if vitamins are great and more people take vitamins, then why they're worse outcomes? So if you only give drugs to doomed individuals, there will never be any effect. So the whole idea of clinical trials is because if you do randomization, you will remove these unwanted random associations between, say, patient-level confounders and outcomes. So if the subjects are randomly assigned to treatment with non-zero probability, then the causal effect is equal to the association. And here's, it's a two-line quote-unquote proof in basic probability. So you want to compute the averages. So this is the average effect if you treat everyone, and this is the average effect if you treat no one. Now, if you randomly selected people to assign into treatment or not, then whether or not there was this symbol here, X, flipped pi, doesn't depend on C naught and C1, exactly means that you have random assignment. If you have random assignment, then you can condition on something you measure. So this is now the expectation of an outcome on people that you treat minus the expectation of the outcome on people you don't treat, which is something you can measure. And this ability to basically say that if I only look at people that I treated, they're fully representative of the entire population, you can only say that if you randomly chose them. If you somehow, allowed people to self-select, or you chose people based on some predetermined category that might be influential in treatment, this fails. So in other words, to summarize, what we've done before, the whole reason you're doing these very expensive randomized clinical trials is to be able to interpret the association that you see, that you can measure, as causation, because by design, the trial it's set up in a way that random confounding is not able to mess up your result. Now, I just want to say that you can build a career, and many people have, on designing clinical trials. It's a very active and complex area of biomedical and biostatistical research. And in addition to a very simple, two-arm clinical trial, there's a whole lot of much more complex and advanced trials in situations where you cannot do full randomization, or it's too expensive or it's too slow. In fact, even in this study, it's not a straightforward, two-arm clinical trial. There were a few other things that they looked at. So we'll see. So this is a study that came out just a few weeks ago in the New England Journal of Medicine. It's a final report on this trial that ran earlier in the year, remdesivir for the treatment of COVID-19. So the goal of the study, to evaluate the clinical efficacy and safety of putative investigational, right? So the keyword here, it's not approved, it's investigational, therapeutic agents among hospitalized adults with laboratory confirmed COVID-19, with designed an adaptive platform trial. So this describes the statistical framework that the trial takes. Well, we'll sort of see what it does. To rapidly conduct a series of phase 3 randomized, double-blind, placebo-controlled trials. Right, so randomized means that people were assigned into treatment and control at random. Double-blind means that the assignment was known neither to the patient nor to the physician. Right, you can have single-blind where the patient doesn't know, but the physician does know. In this case, the double-blind removes another possible bias that could be caused by, say, the change in treatment that would ensue if the physician knew that the person was receiving remdesivir. Placebo-controlled trials, right? So in this case, you're not comparing two drugs, you're comparing a drug to a no drug. So here we describe the first stage of the adaptive COVID-19 treatment trial, abbreviated to ACTT1, in which we evaluated treatment with remdesivir as compared to placebo. So as a reminder, clinical trials and drug approval goes through a series of stages. So here's a useful cartoon for drugs. We have phase 1. So there's a preclinical phase, which is not shown here, where drugs are developed and tested in animal models, and that takes a long time. So we were lucky, if that's the right word, with COVID-19, because there were drugs developed for the original SARS-CoV-2 that went through the preclinical. So we were able to basically skip a lot of the original development and just try them right away and go to phase 3. So phase 1 is test drugs in healthy individuals, and the purpose of that is to look for safety, dosage, and side effects. So this is just a due diligence to make sure that they're well tolerated. Phase 2 then goes on to test on large group of affected individuals. The goal here is to test for efficacy and side effects. Assuming that this succeeds, then we'll move on to a larger phase 3 trial, where you now do new and wider demographic, and you would test here for long-term effectiveness in comparisons with other medications, if there are any other available. And then upon completion, successful completion of phase 3, the drug in the US receives FDA approval, which means the treatment was determined effective and safe for public use. Typically after this, you have subsequent trials, not mandatory, but they might, which basically continue to test for effectiveness and safety. And if unappreciated or unknown side effects or adverse reactions are uncovered, then the approval can be revoked and the drug can be taken off the market if necessary. It does occur. Not frequently, but even in the last 10 or 15 years, there were a couple of fairly high profile revocations when drugs had to be taken off the market. So what is remdesivir? So there's a link up at the top of the page. If you want to, you can go to this drug info website at the National Library of Medicine to find all the information about remdesivir. So remdesivir is a direct acting antiviral agent that was developed by the pharmaceutical company called Gilead. As of today, it has been approved for COVID-19 treatment in several countries, but not in the US. For example, it's approved for treatment in Australia and a few European countries. It was originally developed to inhibit one of the key enzymes in the coronavirus replication cycle. So single-stranded positive sense RNA virus have RNA-dependent RNA polymerase. So I think in lecture 2, we discussed this at great length. The virus has to make lots of full and partial copies of its RNA, so it can be translated into protein and then further packaged into new virions. It was originally developed in the context of SARS-CoV-1 and MERS. The reason it was even considered for trials in humans is because there was a lot of preliminary information. It was shown to inhibit SARS-CoV-2 in vitro, and it also showed reduced viral levels and lung damage in non-human primate models in MERS coronavirus and other animal systems. So this is preclinical information that is essential. So here's an interesting side story. I found this letter that was put out in June by the CEO of Gilead that described sort of their business rationale for pricing the drug, what it was, and it's worth sort of reading in its entirety. The letter is long, but there are just 2 paragraphs in it that I think are interesting. So in normal circumstances, we would price the medicine according to the value it provides. The first results from the NIAID, which is National Institute for Allergy and Infectious Diseases, a part of the NIH, in hospitalized patients with COVID-19 showed that remdesivir shortened time to recovery by an average of 4 days. Taking the example of the United States, earlier hospital discharge would result in hospital savings of approximately $12,000 per patient. Even just considering these immediate savings to the healthcare system alone, we can see the potential value that remdesivir provides. This is before we factor in the direct benefit to those patients who may have a shorter stay in the hospital. We have decided to price remdesivir well below this value to ensure broad and equitable access at a time of urgent global need. We have set a price for governments of developed countries at $390 per vial. Based on current treatment patterns, the vast majority of patients are expected to receive a 5-day treatment course using 6 vials of remdesivir, which equates to $2,340 per patient. There is, right. So it's, it's expensive, but as far as, U.S. Medications go, this is a bargain because, for example, there's certain cancer drugs that will, cost 20 times as much. But the question of ethical, great question. Do you know how much remdesivir costs to manufacture? It's a great question you asked. Somebody calculated the cost about $1 per vial. So, but it's, so you can take it two ways, right? Obviously, Gilead is going to make a whole lot of money on it until there is something else on the market. But I always, want to caution, against making inferences that this means that they're making $380 or $389 per vial, because you always have to factor in the immense sunk cost that goes into drug development. So the cost of manufacturing itself is very cheap, but I do not know the number of, if you do R&D and other, sunk costs, what it would be. But I'm sure they're still making profit on it. And, considering how many people are likely to, receive that treatment, it's a very, very lucrative drug for Gilead. And so now I'm going to spend about 10 minutes or so setting up, explaining to you how this trial was set up, because a hallmark of a well-run trial is that it's going to be very, very diligently and meticulously planned out. In fact, it has to be done prior to its approval. And the other thing that has to be done or has to be very carefully considered is that you basically lay out the entire plan for the trial before you start it. And only under unusual circumstances do you change this, because changing it in the middle of the trial could introduce a bias that you don't appreciate. In fact, this is kind of a unique trial because they did change one of the measures in the middle, and they explain, why it was done. So, so this trial enrolled patients between the end of February in 2020 and April 19th, 2020. So as you can see, it concluded quite a while ago. It is a very large-scale trial. I would not guess how much it costs to run, but tens of millions of dollars is a lower estimate. Maybe not tens of millions, but millions of dollars for sure. With this many trial sites. So another thing to keep in mind: these clinical trials are always very involved and very expensive. It included 60 trial sites and 13 subsites that were spread internationally, but mostly based in the United States. So 45 sites in the United States, 8 in Denmark, 5 in the United Kingdom, 4 in Greece, 3 in Germany, 2 in Korea, 2 in Mexico, 2 in Spain, 1 in Japan, and 1 in Singapore. Whenever you run this trial, any trial, you have to declare very clearly who's eligible to participate. Right. So in this case, there were COVID-19 patients that were admitted to hospital. So among eligible patients that enroll at a particular site, that were randomly assigned in a 1-to-1 ratio. That's not always 1-to-1. Sometimes it's 2-to-1. Sometimes it's 3-to-1. But typically it's 1-to-1 to receive either remdesivir or placebo. One of the first kinks is that they were stratified by study site and disease severity, which means that the random number that was drawn was not drawn across the entire world, but they were divided in one ratio in each site, right? So if we're 50 people, say, enrolled in Korea, 25 of them would be, randomized to placebo, and 25 of them would be randomized to remdesivir. And in addition, they also made sure that people that have different, that came in being somewhat sick, very sick or terribly sick, and it's also been quantified, are split into this one ratio according to the disease stage. And they defined severe disease as one of those things, like requiring mechanical ventilation, requiring supplemental oxygen, low oxygen saturation as measured, by pulse oximetry, or tachypnea, so high respiratory rate. So basically shortness of breath defined precisely. Next, they have to declare how they're going to deliver the drug. So how many doses, how often, what volume. So remdesivir was administered intravenously or intravenously. It was an original 200 milligram loading dose on day 1, and then there were 9 subsequent doses that were half that, so 100 milligrams daily on days 2 through 10, or until hospital discharge or death. Remember, they want to keep this double blind, so they want to they don't want the doctors to know what they're giving a patient. So whenever possible, they would make a matching placebo, so identical packaging, identical color, identical everything. It would be administered exactly the same way in the same volume as the active drug. However, because this was sort of an accelerated scale trial and it involved many international sites, they make a note that sometimes a normal saline placebo was used owing to shortage of matching placebo, in which case they made a specific provision to make sure that the drug and the placebo were indistinguishable. So were masked with an opaque bag and tubing covers to maintain blinding. So very important. And whenever a trial is run, it's in addition to whatever normal care they would receive, right? So a person still goes into the hospital to be treated for COVID-19, which means that the physicians will make every effort as appropriate to what is known to help them recover. So whatever else they would have normally gotten, fluids, bed rest, mechanical ventilation, all of those things, they would still receive regardless of whether or not they were getting remdesivir or not. So this is also very, very standard in these types of trials. Now, protocols were approved. So this is, all the trials have to go through, all the clinical trials have to go through a rigorous review process by special organizations called institutional review boards, or IRBs, in the United States, and they're typically run by a hospital or a university or whatever clinical site. So they were all, individually administered. Now you might imagine, this is being run at 60 different sites, so potentially that means you have to get 60 separate approvals. Not quite as many, because sometimes they can be centralized, but nonetheless, there's a lot of paperwork and administrative oversight involved, which adds complexity and rigor. Study is also overseen by an independent data and safety monitoring board. All right, so for especially studies of that magnitude, you need to have people that are not involved, they're not interested or not entangled in any way, to provide independent data and safety review. So they have to sign off on whether or not the process is going as planned and they're following all the rules. And written informed consent was obtained from each patient or from the patient's legally authorized representative if the patient was unable. I would encourage you to actually, if you've never seen one of those, at the end of the lecture, I have a link to the second or the third stage of this trial where they're doing something else that's still ongoing. You can actually look at what an informed consent form is like. If you ever participate, I'm sure you've signed some form of at some point in your life, in a physician's office. But these are quite long and interesting because you can see, how risk and benefits are communicated to a person, right? They need to be able to communicate, sort of an informed consent has to be written in a way that makes it very clear and understandable, what the trial is doing, what are the potential benefits, what are the potential risks. So a person that, is either is consenting is able to do it with an understanding of what they're consenting to. All right, now, when you run a trial like this, or any trial, you also have to be very precise about what it is that you will measure, and how you will assess success or failure. And this all has to be defined beforehand. So here, for instance, the protocol was the patients were assessed daily during their hospitalization stay. So day 1 through day 29. Obviously, not everybody stayed until day 29, but they basically stopped. If somebody remained in the hospital past day 29, that would be basically stopped for the purposes of this trial at day 29. So then they define a clinical status, defined in an 8-category ordinal scale, which I'll show you in the next slide, and one of the standardized scores that is used for recording the general state of health of an individual. So National Early Warning Score, which includes 6 physiological measures that's recorded daily. So this National Early Warning Score can have a range from 0 to 20, and higher score indicates greater clinical risk. Categorical or numeric scale like this are very common in hospital settings because they standardize care and assessment. So you can directly compare things without having to read texts, and it helps physicians sort of categorize things and avoid making mistakes. And then all serious adverse events and grade 3 or 4 adverse events that represented an increase in severity from day 1 and any grade 2 or higher suspected drug-related hypersensitivity reactions were recorded. So you need to know this to assess the safety of the drug. And this is the key measure of success. So every trial will have at least one primary outcome, which is exactly what they're measuring. And this is primarily what the study was designed to test. So whenever you see, reference to a clinical trial, in the back of your mind, you need to ask yourself, what was it that they actually measured? And for most trials, it will be very, very precisely defined. So for this trial, it's defined as the first day during the 28 days after enrollment on which a patient met the criteria for the category 1, 2, or 3 on the 8-category ordinal scale. So here's a scale where 1 is basically healthy, not hospitalized, and no limitations of activities. 2 is, home recovery kind of thing, so released from the hospital with some limitations of activities, possibly a home oxygen requirement or both, and sort of up the rank with sort of increasing severity. Hospitalized, not requiring supplemental oxygen, and no longer requiring medical care. For example, used if hospitalization was extended for infection control or other non-medical reasons, for example, quarantine. Hospitalized, not requiring supplemental oxygen, but requiring ongoing medical care, and, all the way up to death. You can also see that, by definition, the patients had to have come at category 4 or higher, because as soon as they go into category 3, 2, or 1, they've basically recovered. So all the patients that were in the study were at best in category 4, right? And obviously not dead. So, 4 to 7 is where they entered. And the idea is that if the treatment's working, you're going to progress from a higher number to a lower number. And the first day, you reach number 3, 2, or 1, you've considered to have recovered. And this is what they're measuring. So you can see, this is set up to basically measure the speed of recovery between people that get remdesivir and people that don't get remdesivir. And most clinical trials, because they're so expensive to run and so challenging logistically, will record a bunch of additional outcomes. So there's about 6 or 7 in this trial, which I haven't enumerated all of them, but just give you some sense. The key secondary outcome was clinical status at day 15, right? So, halfway through, instead of 28, day 15. And then many other secondary outcomes, a time to improvement of 1 category and of 2 categories from the baseline ordinal score. So not just going to, 1, 2, or 3, but say going from 6 to a 5. Mean change in status on the ordinal scale from day 1 to days 3, 5, 8, 11, 15, 20, and 29. And then the incidence and duration of new oxygen use, non-invasive ventilation, high flow oxygen, and so on. So lots of things that they've thought about could be informative about how effective the treatment is. And you also have secondary safety outcome measures. So how often did they see, various adverse effects? And grade 3 and 4 are also precisely defined. discontinuation, temporary suspension of infusion. So when did if you had to stop giving the drug for any reason, or some dramatic changes in, say blood work or other laboratory values over time. And then you also need to declare beforehand, which statistical methodology you're going to use to assess significance. And it has to be done ahead of time, again, to avoid bias or confounding. So in this case, the primary analysis was something called a stratified log-rank test of time to recovery with remdesivir as compared with placebo, with stratification by disease severity. So actual severity at baseline. If a patient did not recover or died, then this would count as not recovered by day 29. It was conditioned on how sick the patients were at baseline, how long did it take for them to recover with and without remdesivir. And they've also pre-specified that they're going to, do the overall analysis and also analyses splitting patients by sex, baseline disease severity, age, race, ethnic group, duration of symptoms before randomization, site location, the presence of coexisting conditions. Now, here's an interesting caveat, which you don't see very often. So originally, they were planning to compare status at day 15. So the primary outcome would have been day 15, not day 28. And they changed it in March because of what they call evolving information. And the evolving information was basically that COVID could last longer than 15 days. So when they set up the trial, the assumption was that, in 2 weeks, you'll either recover or die, or in some other way you will show response to treatment. But they basically, by watching cases that were accumulating, case reports and other trials, they realized this might not be long enough. So they proposed the change in March 22nd after 72 patients had been enrolled. And the change was proposed by trial statisticians who did not know, which patients were assigned to which arms, and they had no knowledge of outcome data, right? So it's not like they looked at the data and said, it looks like the trial's failing, so let's change the rules. And then importantly, they also recorded the initial primary outcome. So, you can also look at this trial and see what it would have shown had they not changed that categorization. And there's another interesting, kink in that trial, in that it was terminated early because it was successful. This is basically, it doesn't happen very often. So when you run these trials, you have a schedule, a periodic schedule, when preliminary results on trial data and safety are reviewed. So it's called an interim analysis. And they did analysis on April 27th of 2020. So pretty much, just a week after the enrollment had completed. And at the time they had recorded enough data, so 482 recoveries and 81 deaths, and they decided this was enough data for statistical purposes to decide if the trial was successful or not. And at the time, it was apparent that remdesivir was significantly better than placebo, at which point the Data and Safety Monitoring Board recommended the preliminary primary analysis report and mortality data be provided to the trial team members from the National Institute of Allergy and Infectious Disease, and they very quickly made them public. And in early May, based on these results, the FDA granted what's called an emergency approval for compassionate use of remdesivir. So it could basically go into clinical practice, as an option. And then another thing that could happen if something like this occurs in a clinical trial is that at that point, physicians that are running patients that are still in the trial could request to identify whether or not people under their care are in placebo or remdesivir. And if they're in placebo, they can switch them over to remdesivir because now, they've assessed that it's safe and efficacious. So it's no longer ethical to withhold what is known as successful treatment. Now, there's a nice flow chart in the study that shows you, what happened at various stages in this trial. So as people, at the very top, there were 1,114 people that were assessed for eligibility. So they were basically viewed as potential trial participants. At this point, a small number were excluded. So 24 of them simply said, I don't want to do it. And 28, after the trial staff reviewed perhaps their health records or some other information, they were declared ineligible. And this is also very formally defined. So there's a list of criteria that you have to meet. You have to be, say, a certain age. You have to be able to consent, or somebody has to consent for you. You need to have a bunch of, you have to meet a bunch of criteria. So, after this initial sieve, 1,062 underwent randomization. So about half and half, right? The reason it's not precisely half and half is because it was, remember, this was stratified by site. So 541 did receive, were assigned to receive remdesivir, 521 did. And then, there's a little dropout here. If you look at the supplementary information, I'm sure I'm going to tell you what happened to them. Maybe they dropped out at this point. And then, as you drill down the line, you can see, how many of them completed each stage of the trial. So, for example, here, 208 received all 10 doses, 323 received fewer than 10 doses, and now they're saying why, because 223 of them recovered, 15 died, 52 had to discontinue because of adverse events, 4 withdrawn by investigator, 6 withdrew from the study, 4 withdrew and transitioned to comfort care, which means they were basically, palliative care. 1 was transferred to another hospital, and so on. And then, 517 completed the study. And finally, this is what was analyzed. So the 532 were included as treated population, 516 were included in as treated population here. And there's, intent to treat versus as treated as a sort of a subtle statistical distinction, because these patients include those that were actually treated and those that were you planned to treat but you couldn't complete based on whatever reason. So sometimes you'll do both analyses to make sure that there's no difference between these two trials. Now here's the table 1, which in any clinical study will include demographic information about the patients. And there's one feature here that I would like to draw your attention to. So they'll describe all the important categorizations of the participants. So, age, and you can see because these were individuals with COVID-19 that had at least some sort of, that required hospitalization, they tended to be older than the median age. So, the mean here is close to 60 years. There were slightly more, actually about 2/3 of them were men. And again, this represents who went into the hospital. You can see that there was a reasonably good representation of various races and ethnic groups. So whites, blacks or African Americans, Asians, and American Indians or Alaska Native, and Hispanic or Latino, which is recorded as a separate category. You can see that they all had about 9 from initial symptoms to randomization. And they're trying to report all the things that might, be confounded with the treatment. So did they have coexisting conditions? No, 1, 2, or more. If they did, what were they? Type 2 diabetes, hypertension or high blood pressure, obesity. And then, how bad was their condition when they entered the hospital? So, I remember I mentioned that they had to be between 4 and 7 by design. So they are 4, 5, 6, and 7. Now, one of the things you want to notice in a well-designed study, if you look between remdesivir and placebo, there should be really close proportions for each of those categories. So like there shouldn't be a systematic difference in age. There shouldn't be a systematic difference in, gender or representation by, race and ethnic groups. So this is nicely balanced for all these categories, which is a byproduct of randomization. Right? You might imagine, for instance, that a badly designed study would take all the people that have type 2 diabetes and hypertension and put them in the treatment arm, and put people that have no pre-existing conditions into the control arm, which might actually make it look like the treatment is doing poorly because you're not controlling for these conditions. By doing randomization, not only do you control for the conditions that you know about, but you also control for all the things that you don't know about. So there might be a lot of other things that influence your susceptibility to COVID-19, and we just don't know about them. So we couldn't control for them ahead of time, but by randomization, we don't bias things one way or the other. And this is the primary figure. So this is sort of, if you were to summarize the entire study in one chart, this would be the chart. This is a primary outcome, and this is a a survival curve. So what you see is also nicely shown here for it's broken down to two arms. So remdesivir or placebo. You follow people over time. So 0 is the day they entered the study. And 29 is as long as the study ran. And for each day, you track what fraction of them recovered. So, at the beginning, everybody's sick. Then you can see that, immediately in day 1, somebody in the remdesivir arm already recovered, right? And then as they progress more and more and more, a larger fraction of individuals recover. If you run it all the way to the end of the study, about 3/4 of everybody enrolled that was in the remdesivir arm recovered, and looks like about 2/3 of everybody in the placebo arm recovered. The key thing to look for is whether or not these curves are different. So if remdesivir is effective, people on remdesivir will recover faster, and more of them will recover at all. So as you look at this curve, for pretty much any day, a larger fraction of people with remdesivir have recovered than with placebo. You have to wait a couple of days for the separation to become obvious, but after about day 5 or 6, they never meet again. So remdesivir is always better. And here you can see the actual numbers of people that were still at risk, right? So this is basically the residual, right? So 541 were at risk in the beginning, and then the end 84 remained. And then placebo 521 and 105 remained. And this is the record for every day. So this is the log rank survival type analysis, which is what they said they were going to do in the beginning. There's a statistical test that you can run that tells you how significant this difference is. I mean, this is pretty obviously significant to the eye. And this is usually good enough to decide that something is effective. So the formal statement of this would be patients in the remdesivir group had a shorter time of recovery than patients in the placebo group. So a median 10 days versus 15 days for placebo. So if you remember the letter from the Gilead CEO, they said an average of 4 days. So this is about right. And the rate ratio for recovery was 1.29, right? So, for each 100 people recovered in the placebo arm, 129 recovered in the remdesivir arm. And you can, because this is a statistical measurement, it comes with error, so you can estimate a confidence interval on it, which will be between 1.12 to 1.49. Importantly, doesn't overlap 1. 1 would be there's no difference between remdesivir and placebo, and the p-value for this is nice and small. So this is a significant result, and this is pretty much the take-home message of the study. So this was, a very, very expensive figure to generate, if you think about it. And it is also a very, very common type of analysis that you will see in clinical trials. This is survival or recovery analysis, where you have 2 time series, and you want to see if they're separated. Now, because they clearly thought about, what might be different between groups, they then went on to split the analyses between groups based on how sick they were when they came in. So in this sense, if you think about it, this goes back to my example of, people that can be survivors, versus responders, anti-responders are doomed. So you might think that patients that are not receiving oxygen, meaning they came into the hospital in better shape, might be less benefited by remdesivir. And you can see that it's actually kind of looks that way, because if you only look at a subset of individuals that are not receiving oxygen, so this is not the entire population, but only about a sixth of it, remdesivir is still better, but it's not as much better. And in fact, the curves no overlap, so they're not significantly different. Now, patients that are receiving oxygen definitely benefit from remdesivir more, because the curves are now separated again. But when you look at patients that are receiving high flow oxygen or non-invasive mechanical ventilation, it is now, you have a different trajectory. So now basically, if you have to, they're sort of different behavior. Where a fraction of individuals recover, but the overall rate of recovery is indifferent. So basically what this tells you is that if you do recover, then you'll do it faster with remdesivir, but there's not a big difference between how many people remain at the end overall. And if you're very sick, means that you're receiving mechanical ventilation, there's no difference. So remdesivir is pretty useless in this context. And here's another really nice summary of what remdesivir does for you, stratifying everybody by various demographic and other characteristics. So here's all the categories you could consider. Here's the number of patients in each category. So 1,062 overall, and broken down by individual groups. And here's what remdesivir is doing for you, depending which category you are. So this is the recovery rate ratio. So ratio of 1 means it doesn't matter if you receive remdesivir or don't, you get the same recovery rate. What you want to look for is you want to look for the rows in which remdesivir is better. So the point is what the estimate that came from the trial, and the bars around it are the confidence intervals. So our degree of uncertainty, because this is a statistical procedure, everything comes back with error. So you want to see for points, look for points that are to the right of 1, and you also want to look for situations where the bars don't overlap the dotted line. So you can see, sort of scanning down the line, that all patients on average benefit from remdesivir. All regions benefit from remdesivir, but because there's not a whole lot of individuals in the Asian and European sample, we're not 100% sure that this is not a statistical artifact. So we need to enroll more people. So see, all the points are to the right of 1, but because there are only 163 patients in Europe and only 52 in Asia, we're not 100% certain. There is a chance that this could just be statistical noise. And the same is true for stratification for race. So all of them look like, they are, the remdesivir is better, but because there is, there's 566 white participants versus 226 black participants who don't have enough statistical power to say that it's definitively above 1 for blacks or for Asians. The same for ethnic groups, the same for age groups, beneficial for both men and women, beneficial for early and late infection, even though it overlaps a little bit. And pretty much the only place where it is completely ineffective is if you come into the study already requiring mechanical ventilation. So, at that point, giving somebody remdesivir doesn't really help them. All right, so now I'm just going to go back briefly. This might take me a while to scroll back, but there was so here. So for example, here you may see that the standard recommendation is, do not give people remdesivir unless they've recently been intubated and doses and durations discussed above. And see, this is not a strong opinion, right? This is only an optional recommendation. So now, and this is actually based on the trial that we just discussed, which is how you get these kinds of gradations. They're just not, they're not guesswork. They're based on outcomes of clinical trials. All right, let me scroll back to where we were. So here's another way to look at these data. And I'm not going to spend it too much, but spend too much time on it. they can basically look at it and digest and kind of, pull out, specific type of outcome that you might want, right? So, for example, what's the overall number of recoveries between remdesivir and placebo? What's the median time to recovery? And you can try to see, breaking down, for example, if you come into the hospital in category 6, which is requiring ventilation, but not mechanical invasive ventilation, then with remdesivir, you take a median of 15 days to recover, and with placebo, you take a median of 20 days to recover. Mortality over the entire study period, right? So you can see, and this is the hazard ratio. Smaller means remdesivir is better. So for the overall population, it's 0.73. But the best bang for your buck with remdesivir you get if you come into category 5, right, where the recovery rate is dramatically better and death rate is dramatically better with remdesivir. So only 9 deaths by day 29 versus 25. And then this is the overall score at day 15. So this was their original outcome. So, score 1, remember, is a discharge from the hospital with no restrictions. So for example, if you look at group 5, 90%, 90 individuals, or 38% of all those that came in at stage 5 were discharged with no restrictions from the hospital, and so on. So this is sort of a detailed breakdown of what the clinical trial did. And the summary, and there's the secondary outcomes. So the odds of improvement in the ordinal score were higher in the remdesivir group. For example, the odds ratio was 1.5 that you improved. There was at day 15, there was a 6.7% rate of mortality in the remdesivir group and 11.9% in the placebo group. So the hazard ratio of 0.5, and at day 29 was 11.4 versus 15.12, which is another thing you might stop and think about it, right? So this is a very, very successful trial, right? And it was, in fact, so successful that it was stopped early, and it was recommended that remdesivir be given to everybody in the trial because it's so successful. But it doesn't prevent a substantial fraction of individuals from dying. It lowers this fraction significantly, but it doesn't drop it to 0. And the secondary outcomes, that patients in the remdesivir group had a shorter time to improvement of 1 or 2 categories, a shorter time to discharge. If they were receiving oxygen, they continued to receive oxygen for fewer days, and there are several others that I don't list. There's another way to break down the differences. For instance, the length of hospitalization. With remdesivir, the median was 12 days. Without, it was 17 days. So the average is 5 days fewer. Median duration of initial hospitalization among those who did not die. There's a fraction of people who were rehospitalized, oxygen, and so on. So very, very detailed breakdown. You can sort of stare at it and try to extract whatever information you want. And then there's also the consideration of safety outcomes. So, perhaps remdesivir helped people to recover from COVID, but they might have acquired some other undesirable side effects. And in fact, so this trial will record serious adverse events in all patients from all causes, right? So whatever it's, it could be something like, depending on what they were recording, it was all stipulated in the trial. An adverse event could be anything from, I don't know, internal bleeding to, say a cardiac episode or anything else that's defined. So they will record all of them. And some of them will be due to remdesivir, and some of them will be due to just being in the hospital or being something else. So they have to kind of separate them. So in a quarter of the subjects in the remdesivir group, there were serious adverse events, and about 31% in the placebo group, right? So this combines adverse events from all causes, right? I mean, these are pretty sick people that are in the hospital being treated for COVID. And then grade 3 or 4 adverse events occurred during the study, in about half of them, half of the individuals in all the groups. And then the investigators review all of these events and then try to decide, again, based on well-defined criteria, whether or not they could be attributed to remdesivir or not. So 41 events were related to remdesivir and 47 to placebo. And some of the common side effects would be like kidney and liver issues, decreased hemoglobin level, a decreased lymphocyte count, respiratory failure, anemia, pyrexia, which is just fever, hyperglycemia, just too much blood sugar, increased blood creatinine level, which is a kidney issue, an increased blood glucose level. The question is, would it be possible to discuss the criteria behind defining respective grades of adverse events? I can look it up for you. I don't have it right for me. This is supplementary information. It's going to be a very, very long list, and it's very standard. So they're just hospital and child documentation. It's basically there's a long catalog of what they are. But here's some of these events that are in grade 3 or 4. So the conclusions of the study is that the Food and Drug Administration issued an emergency use authorization on May 1st, so less than a week after that interim study, to permit the use of remdesivir for treatment in adults and children. Remdesivir has also received full or conditional approval in several other countries since that time. However, as I mentioned just a few slides ago, it's clear that remdesivir alone is not sufficient for all patients, because a substantial fraction of people don't recover, and a substantial fraction of them go on to die. So as a follow-up, there are hundreds of different clinical trials being run, but at the moment, remdesivir is being evaluated in combination with modifiers of the immune response. So there are 2 subsequent trials called ACTT2, where remdesivir is coupled with a JAK inhibitor called, baricitinib. I think it's a cancer drug, but don't quote me on that. And interferon beta 1A in ACTT3. So it's combination therapy. And this is also something you see quite often, where you basically build a regimen treatment where you use compounds and treatments concurrently with the hope that they enhance each other, and they sort of build that up. And typically you would run separate trials for all of them, using outcomes of previous trials to justify subsequent trials. But obviously you need to have some information, before you run this. For example, if you're on interferon beta 1A, you need to have some evidence that interferon beta 1A, would be better in combination. Right? So there had to be some in vitro or animal model studies. And then a variety of therapeutic options, including novel antivirals, modifiers. So novel antivirals would be something developed specifically for SARS-CoV-2. Modifiers of the immune response or other intrinsic pathways. Combination approaches are needed to continue to improve outcomes in patients with COVID-19. So this is, what I mentioned a little while back. If you want to go and, for example, look at, what a trial that's ongoing looks like. Here's the follow-up to this trial that's available in clinicaltrials.gov. It's Adaptive COVID-19 Treatment Trial, or ACTT2. You can see that this is now, it's concluded enrollment. So it's still running. So first posted May 26th, last updated April 13th. You can see what it is. We'll evaluate the combination of baricitinib and remdesivir compared to remdesivir alone. So I can already tell you that this is now not going to be comparing to placebo. We're comparing combination remdesivir to remdesivir alone. And it's going to tell you exactly what they're going to do. So it's a very similar setup to what we just discussed. Primary outcome is time to recovery by day 29. And there's the same record for ACTT3. And there's a bunch of other clinical trials. So, I would encourage you to go and browse it and see what you can find. All right. Are there any questions at this point? Because I'm going to switch gears and talk a little bit about something other than SARS-CoV-2. And then we'll come back to discussing immune therapies and other things that people are trying next time. All right. So seeing none, I will continue to the second part of the lecture, which is you're going to switch gears and talk a little bit about bacteria. So we spent obviously a lot of time talking about viruses, so bacteria and viruses are both microbial pathogens. Bacteria are bigger than viruses and different in many different ways. So let's sort of identify what the differences are. So bacteria is definitely a living organism. Virus, it really is difficult to determine if it's alive or not because it's not capable of independent replication. So by that definition, it's not alive, but as sort of a reproducing organism that is selfish and wants to, continue existing, it is very much alive. A bacteria is a unicellular prokaryotic organism. Viruses have no cells, they're viral particles. Bacteria have DNA and RNA in cytoplasm. It has a well-defined cell wall and membrane. The physical construction of a virus, it has a DNA or RNA payload enclosed inside a viral particle. The cell wall is also different, so you have basically peptidoglycans, so sugars, and lipopolysaccharides, fats and sugars, on cell wall in bacteria, and protein viral coat, which is specialized viral proteins. You treat bacteria with antibiotics. Antibiotics don't do anything against viruses. Viruses are traditionally treated with vaccines and direct-acting antivirals. Bacteria have enzymes. RNA viruses don't have enzymes, but some DNA viruses do. There's no nucleus in either. They're both virulent. Bacteria typically causes localized infection. So something like, the site of infection is where it's replicating. Virus is typically systemic, so it gets into your bloodstream and spreads to all parts of the body. Bacteria reproduces by fission, which is a form of asexual reproduction, and virus cannot reproduce independently, but it uses a host cell to replicate, eventually killing the host cell. They're very different in size, so bacteria are about a micron in size. Viruses are 1 or 2 orders of magnitude smaller physically, and similar is true for genomes. Bacterial genomes tend to be between 1 and 10 megabases, and viruses are between 10 and 100 kilobases. SARS-CoV-2 is at 30 kilobases. Here are some examples of famous or infamous bacteria. So you have E. coli. Here's the whole family tree or categorization of where bacteria E. coli is. So it belongs to gamma proteobacteria. Bacteria tend to get classified sort of the highest level into 2 bins. So gram-negative or gram-positive. So gram-negative has a dual membrane, gram-positive has a single membrane. E. coli has a fairly average bacterial genome, 4 to 6 megabases on a single chromosome. Depending on the strain of E. coli, it will have plasmids, 0 to 3. There are about 5,000 genes in a bacterium. If you recall, a typical virus has something in the order of 10 to 20 genes, and there's a circular DNA. E. coli mostly causes urinary tract infections, neonatal meningitis, and I'm sure the first thing came to your mind, it's intestinal disease. The mechanism through which it acquires its pathogenicity, usually by attaching to the host cell and introducing toxins that disrupt normal cellular processes. So very different from viruses. Viruses actually break up your cells. Bacteria basically release localized poison that messes with your cells. Not all E. coli, most of the E. coli are not pathogenic, they're benign, but the ones that are pathogenic, typically, a lot of times they get their pathogenic proteins on extrachromosomal plasmids, or within bacteriophages, which are viruses that infect bacteria, and distinct DNA segments turn pathogenicity islands. So plasmid is basically an independent circular double-stranded DNA molecule, which is distinct from chromosomal DNA. They're typically much shorter in length, from 1 to 100 kilobases, and they naturally exist in bacterial cells and some eukaryotes. Importantly, they're copied during bacterial division. They can migrate from bacteria to bacteria. So you can think of plasmids as basically DNA payload delivery mechanisms, where bacteria can borrow genetic information from other bacteria. A few other examples of common bacteria would be Staphylococcus aureus or Staph A. This is gram-positive. This most commonly causes boils and furuncles and other superficial skin infections in humans. also enzymes and toxins. The interesting thing about Staph aureus, and we mentioned this a couple lectures ago, we're talking about B cells, is that it includes superantigens which cause the toxic shock syndrome, salmonella, Mycobacterium tuberculosis, and then, since we mentioned hydroxychloroquine, I'd mentioned Plasmodium, which is what was designed to treat, which is not a bacterium, it's a protozoan parasite that causes malaria. So one of the things about bacteria is it's probably the most abundant domain of life. There's a huge number of bacterial species that we're aware of, and a much larger number of bacterial species that we're not. There are a number of efforts to maintain sort of overall trees of all the known bacterial species. Interestingly, it's difficult to define species in bacteria because they tend to exchange genetic information with one another. And they also tend to lose and acquire genes, right? So it's quite common for bacteria in the same species to only have about half of the genes in common. So they're core genes and they're sort of accessory genes that were not essential for survival. So defining bacterial species is much, much harder. It's imagine, for example, if, that's enough. So so because of that, bacterial trees are usually drawn using a gene that is present in all bacterial species. And this is, typically would be something remarkably conserved that's involved in some basic function like, protein production or DNA replication. So ribosomal RNA, 16S or 23X, are sufficiently conserved to retain homology signal and sufficiently diverged to be informative. Question is, I know the bacterial genomes don't usually have telomeres, but is it possible to introduce the repetitive telomere region present in eukaryotic cells? I don't know. Maybe. I'm sure if you're creative enough, you can introduce anything via transfection or transduction. I'm sure you guys saw the Nobel Prize in chemistry went to two of the primary discoverers of CRISPR, which is a gene editing technique. So I don't know how to do it. I'm not a molecular biologist, but I'm sure it can be done. As far as genomic analysis goes, because after all, this is a genomic course, so I need to mention something about genomics in every lecture. So compared to viruses, bacteria have relatively long genomes with many genes, which makes genomic analysis a little harder. Can bacteria hold viruses? Yes, the bacteriophages are viruses that infect bacteria, and they're very abundant and very lethal. So viruses infect literally everything, except other viruses. But bacteria have much lower mutation rates, which makes their genomes more stable on one end. But on the other, they have what's called frequent lateral gene transfer, which means bacteria acquire genes from other bacteria in the environment. So what I want to sort of lead into and continue discussion in this context in SARS-CoV-2 next time is the idea of antibiotic resistance. And this is important, focusing back on COVID, I can guarantee that if we continue treating COVID-19 with the same drug, that is a single therapy drug that only targets one protein and only has one binding site, we're going to develop drug resistance. It's not going to be pervasive, it's not going to be guaranteed, but there will be drug resistance because that's what pathogens do. They escape the action of drugs. It's happened every time in pretty much every system to different degrees. And the only sort of successful solution we have to this now is combination therapy, where we hit the pathogen with multiple drugs, which makes it very unlikely that it's going to develop a simultaneously resistance to 2 or 3 agents. So antibiotics are, it's a class of drugs that, broadly speaking, neutralize bacteria in a variety of mechanisms. And the introduction of antibiotics is one of the most important medical interventions in the 20th century. And I would say before the COVID pandemic, one of the biggest public health threats was the increasing frequency and breadth of antibiotic drug resistance in bacteria, to the point where there were sufficiently multi-drug resistant bacterial types in hospitals that you basically couldn't treat them with anything, and doctors would resort to sort of holding out a couple of antibiotics as the last line of treatment, and there was a significant push to try to identify new antibiotics. Question is, do you think reducing the amount of drugs prescribed will have reduced antibiotic resistance to bacteria? That's one of the, indiscriminate use of antibiotics is one of the contributors to increased drug resistance. And but that's not the only thing that you can do. So for example, new antibiotic discovery is just as important. Another question, do you believe that exploration of bacteriophage therapies will be effective in addressing the antibiotic problem? It's a great question. So phage therapy is an idea that you basically kill bacteria by treating patients with viruses that kill bacteria. There is there's definitely an increased emphasis in that area of research. So there was, before Temple, I spent many years in medical school at UC San Diego, and when I was there, one of the I was working with infectious disease doctors and HIV primarily, and a few other bugs, but there was a famous case that made the national news, and there was a book written about it. But there was this anthropology professor, Tom Patterson, who he and his wife went to Egypt, and he became infected. He got peritonitis or something like that, some intestinal bacterial disease that turned out to be completely resistant to all antibiotics. So he was flown from there to Germany, then from Germany back to San Diego, put in the university hospital, and everybody thought he was going to die because there was no no effective treatment. So in a lark, one of my colleagues, an infectious disease doctor there, saying, why don't we try phage therapy? And it was actually able, phage therapy worked. He completely recovered and is back to normal now, and is sort of the walking example of how you can use phage therapy to treat very recalcitrant and extensively drug-resistant bacterial infections. So yeah, thank you, Peter, for asking that question. I wasn't going to talk about it, but it's a very promising area of research. So the intensive use of antibiotics, there was the previous question, will we use between 100,000 and 200,000 tons worldwide. So imagine, that's a massive quantity of antibiotics. That's like a train load. That would dramatically increase the frequency of resistance. And resistance severely reduces the possibility of treating infections effectively, increases the risk of complications of a fatal outcome. Question is, why not just target conservative protein regions on the pathogen? You could, but there are a couple of potential challenges. There are actually very few sort of universal conserved protein regions, and most of the antibiotics that we know are basically borrowed from nature. So we took compounds that were developed by other organisms to fight bacteria and repurpose them, and they don't work directly with proteins. They work with something else. So we'll see. So here's a global picture of what the resistant looks like. So this was from a study, meta-analysis of 58 studies, which means there was a follow-up study where they combined information from 58 other studies, urinary tract infections in children between 0 and 17 years old. And here's the global picture of drug resistance separated by countries that are either OECD, so Office for Economic Cooperation and Development. So these are the countries that are shown in blue are members of OECD, and the countries that are shown in gray are non-members. And here you have drug resistance sort of by a couple of antibiotics. Ampicillin is the oldest one, and then ciprofloxacin is, if you've ever seen it, it's usually abbreviated as Cipros. It's one of the stronger antibiotics. And then you have, adrofurantoin. So you have, for common antibiotics, drug resistance basically over 50%. And then non-OECD countries, even the sort of rare, stronger antibiotics are also significantly, significantly impacted by drug resistance. So one possibility is that in many non-OECD countries, you have over-the-counter antibiotic availability, which means that people can self-prescribe and improperly administer antibiotic courses. One of the worst things you can do, and which is why if you're ever prescribed antibiotics, you're told to take the entire, however many pills you were prescribed. And that's because if you're given 10 doses of antibiotic and you start taking it and you take 3, say, oh, I'm feeling better now, I'm going to stop taking it. What that does, it doesn't completely eradicate the bacterium. It sort of knocks it down into submission, but then you stop taking the drug. And if by accident, one of the bacteria now has an opportunity to develop drug resistance and grow back out. Question is, I know this might be a little off topic, but are COVID antibody tests accurate? Great question. There is, I don't remember where. I think it might be on my lecture 3. And if I'm not going to remember it now, but, text me on Slack and I'll send you the link. There is a great website which keeps track of accuracy for all the available antibody tests. So there's basically, I think it's run out of Switzerland, if I remember correctly. So what they do, they keep track of all the literature and other performance tests of antibody tests. So, academic groups and clinics can run, their own experiments to evaluate how accurate or precise a particular test is, and they're reported there. So you can look up by specific test, by type of test. You can look what kind of accuracy it is. But typically, antibody tests have, if anything, they sort of suffer from lack of sensitivity. So they may give you their Achilles heel is that they don't always tell you that the antibodies are there. Whereas QRT-PCR, so nucleic acid tests, depending on how they run, could suffer from too many false positives. But it really depends on the test. And thankfully, there's great information out there that you can use. As far as antibiotic resistance acquisition goes, there's sort of 4 main routes for bacteria. Keep that in mind for viruses as well. Not all of them are available. So for viruses, pretty much the only thing you have is mutation. For bacteria, you have various other things where you can have. So direct mutation is simply you're modifying the gene of interest. Or you could also just borrow resistance genes from other cells. So you can do it by conjugation, where you basically there's this little plasmid that's shown here as carrying the resistant genes. You can have transformation, which is basically cell-free plasmids or linear DNA is absorbed by the cells. Or you can acquire it through bacteriophage infection. So you can actually take pieces of a bacterial virus and incorporate it into your own genome as a bacterium and become resistant. And there are different types of molecular mechanisms of antibiotic resistance. In this case, from gram-negative, where you have dual membranes, you can sort of have natural resistance, which means the drug fails to get inside the bacterium. You have something called efflux pumps, which are these things that sit across nanopores, which we use now for sequencing. You might remember this, repurposed nanopores for sequencing, and they just pump chemicals out of the cell. You can also have direct resistance mutations, where whatever protein is being targeted by the drug changes, so the drug is no longer able to bind. Or you can have something that inactivates the drug itself. Right, so they're just specific examples. You could also look for resistance, try to identify resistance in natural samples. If you know the loci or the genes that are involved in antibiotic resistance, which you don't always do, but if you do, you can PCR amplify them using gene-specific primers, and then do, say, gel electrophoresis to see if they're there. You can also do microarrays and sequencing. If you don't know which genes are responsible for resistance, you can do functional screening, where you take a soil sample doesn't have to be a soil sample. It's extracted and cloned into libraries. Then these libraries are introduced into E. coli or other bacterial host, and then you try to grow them in the presence of the drug to see if they're resistant or not. The question is, how would combination therapy help in a multi-drug resistance bacterial infection if all the bacteria had resistance for all the individual drugs employed in combination? So combination therapy, it's also a very good question. Combination therapy is primarily designed to avoid the development of drug resistance. So you, because you're hitting multiple targets in the same pathogen, it is simply not able to fortuitously acquire, multiple necessary mutations to escape the action of all the drugs. If you have something that's multi-drug resistant, you need to design a custom treatment for it. So you basically need to rationally figure out which drugs you can deploy and which drugs are likely to be effective. And in some cases, there's simply nothing, right? So you have to come up with creative treatments like, bacteriophage therapy. So I'll conclude by, sort of giving you a very brief description of how drug resistance is determined. And in the reading for next time, you will see this, a much more modern version of this applied to understanding drug resistance that SARS-CoV-2 develops to monoclonal cocktails like Regeneron. So it's a really interesting paper there that you guys will read next time. So let's look at streptomycin, which is an old and effective broad spectrum antibiotic. And the reason it's broad spectrum is because it interferes with an essential function in all bacteria. It interferes with protein synthesis in bacteria. Specifically, it binds to or modifies the 30S subunit of bacterial ribosomes, which causes them to misread transfer RNAs and synthesize the protein. So that basically destroys all of the life cycles in the bacteria. So it's an incredibly effective drug that sort of just kills everything that it sees it's effective against. It's an unavoidable function. And it's specific to bacteria, right? Because we also have ribosomes, and we also need to read and properly transfer RNAs, but human ribosomes have a different structure. So this drug has no toxicity to humans. And because it targets an essential process of bacterial protein synthesis, it targets both gram-positive and gram-negative bacteria. And streptomycin was the first successful tuberculosis treatment. And it's a very, very old drug. So first characterized in the 1940s, but pretty much within 10 years of its discovery, people realized there was also resistance to it. So resistance frequently acquired through point mutation, which is what viruses typically do, and this is what SARS-CoV-2 is likely to do against remdesivir and other drugs that we deploy on a broad scale. I'm actually going to mention the paper next time where there's already discussion about how this could happen. Resistance mutation will modify, either proteins or ribosomal RNA, right? So ribosomal RNA works in context with proteins. So by modifying, one or the other, you can prevent the drug from acting effectively. So here's a paper from 1972 that quantified drug resistance using very laborious, very slow throughput sequencing technique. When you did digestion by restriction enzymes and 2D paper fingerprinting. And you must understand that in order for this to work, you basically needed to know exactly where to look. So you needed to know the protein that you were going to sequence. And you also had a good understanding of what the mechanism of escape would be. So this is not something you could, you could do to discover which genes are involved in drug resistance. This you would do if you knew or had a very, very good idea which genes are, and you just confirm. So here so what you have here is a collection of isolates, right? So there are 3 isolates here. So 3 parental strains, and for each of them you have individual isolates. And all they're basically doing is recording this repeated mutation from lysine to arginine that occurs either in position 87 or position 42. Right, so this is a single point. No, there's one lysine to threonine. So you have basically repeated substitution. The question is, how did they know the approximate area where the gene conferring resistance was located? I mean, they knew it precisely for streptomycin because they knew what it was targeting. And then you could isolate on 30S proteins. Ribosome structure analysis was one of the first things that people were looking at in molecular biology. So it's kind of a confluence of two things. It's also very abundant, protein. But you're right, you needed to know exactly what to look for. So it means the ribosomal structure, which is a complex multi-protein mechanism. You needed to know how to isolate the specific protein. But nonetheless, this is one of the earliest direct sequencing based experiments to identify resistance. But here's one of the things you will see why this is so compelling, because you see the same changes occurring in independent strains, right? So you see the same two positions, 87 or 42, and you see the same substitution, right? So lysine is always involved, and then you have one nucleotide change that goes to either arginine or arginine, asparagine or threonine. So this is repeated, and this gives you additional confidence. Drug resistance doesn't always have to be like this. You can have multiple pathways to resistance, but in many pathogens it typically is pretty deterministic. So in HIV, there's sort of very, very well defined drug resistance mutations. In influenza, they're very well defined drug resistance mutations. And if we're going to see something in SARS-CoV-2, I'm sure there will also be a couple of positions that have recurrent changes. So here's stepping up to 1993. So molecular basis of streptomycin resistance in Mycobacterium tuberculosis. So now this alteration within the ribosomal protein 16 S12. So now they do direct sequencing of 73 isolates with Sanger sequencing, and notice that it's the same positions, except in the previous slide it was 42 and 87, and here it's 43 and 88. So they probably just had a slightly different coordinate systems. So you had the same change confirmed in either multi-drug resistance or single drug resistance, and in control isolates there weren't any. And they did some molecular characterization of what these mutations do. And then moving on to what you would do now in modern throughput sequencing. So here's basically a high throughput analysis of a large number of multi-drug resistance isolates just to discover what's there. So this was done in 2013. They did full genome sequencing of 161 bacterial isolates that were confirmed by assays to be multi-drug resistant using the Illumina platform. So approximately 500 megabases sequenced per isolate. And they just cataloged what was there, right? To see if they were all resistant the same way, repeated mutations, different mutations, which genes. And here's an example of how you can summarize these large types of studies. So we haven't seen those in a while. I'll remind you, this is a phylogenetic tree, which shows you the relationship of the different strains. This is whole genome sequencing. So this is over 5 million base pair. This is the difference, the tree. So this is less than 1% distant across the entire genome. So much slower evolving than viruses. And so there's 161 isolates in the study, plus they included 22 reference genomes, which are in gray. So these are known colors, are different lineages of Mycobacterium tuberculosis. They're colored by area of isolation. So purple lineage, red lineage, brown lineage. And that strains labeled in gray represent previously published genomes. Those in blue represent drug-sensitive strains, and those in black represent MDR strains, right? So you can see they have sensitive and resistant strains, and those in pink represent XDR strains. So XDR is extensive drug resistance, so worse than multiple drug resistance. one of the things you can see here is that the colors of the isolates are mixed, which means that, for example, here you see an XDR isolate sitting right next to a susceptible or a sensitive isolate or a multi-drug resistance isolate. What that means is that resistance is a feature that evolves repeatedly in bacteria. If you had all drug resistance mutations sitting on one part of the subtree, that would mean that somewhere in the past, there was one event that made them all drug resistance, and they just inherited it from there on. Because this drug resistance scattered throughout the tree, that means that each of those lineages, each of those bacteria found its own way to become drug resistant. And it's kind of all spread throughout all the lineages, so none of them are specifically too drug resistant or too sensitive. It's sort of a common pattern that bacteria, if you leave them alone and expose them to enough antibiotics, will acquire drug resistance. And here was the summary of where they found on the genome these drug resistance locations were. So there were 85 drug associated genes. They confirmed the genes that were previously characterized, so shown in red, and then they found a bunch of new ones. The blue ones, sorry, there's a few things going on here. So the circle is the bacterial genome. 0 is where it starts. You go clockwise and wind it all the way to the end of the genome. Individual stripes on this circle mean that there were mutations in those genes and multiple drug resistance, extensively drug resistant strains. Red ones were those that were previously known to be implicated in drug resistance. And if you have an additional name, so see some bars have no names to them, and some bars do, like this, cis-N. The name genes are those that are considered essential for the in vitro growth of the reference strain, which means that if you delete them, the bacterium doesn't grow. The others are quote unquote optional. They might have significant functions that the bacteria can survive if you knock them out. So basically mutations in those genes that are named are likely to have more functional significance for the bacteria. I'll skip that one because it's late in the day. All right, that's all I have for today.